The search for the truth began with tragedy. Over 100,000 people had lost their lives to a mysterious illness that spread silently, leaving devastation in its wake. For years, doctors, scientists, and grieving families demanded answers, but the cause of the outbreak remained buried under uncertainty. The disease struck without warning, often beginning with flu-like symptoms. Fever, chills, fatigue, but quickly turning far more severe. Victims experienced organ failure, neurological decline, and a cascade of immune responses that baffled even the most experienced physicians. Entire wards in hospitals were filled with patients gasping for breath, families weeping by their bedsides, and nurses working until collapse. The numbers rose daily, and soon the death toll surpassed 100,000. Governments declared states of emergency, but despite every measure, lockdowns, quarantines, rushed vaccines, no one truly understood what was killing so many. Speculation filled the void. Some experts insisted it was a new strain of influenza, one that had mutated faster than usual and caught global health systems unprepared. Others argued it was a form of hemorrhagic fever, hidden for years in remote regions before being unleashed on cities through trade and travel. Rumors spread like wildfire. Conspiracies emerged, claiming it was a biological weapon, a laboratory escape, or even a government cover-up. Each theory added to the confusion, and the world felt paralyzed by fear. For ordinary people, it hardly mattered where the disease came from. What mattered was that loved ones were dying, and no cure seemed near. In laboratories across the globe, scientists raced against time. Samples of tissue, blood, and spinal fluid were collected from the dead and dying, examined under microscopes, and tested for every known pathogen. Yet nothing matched. The genetic fingerprints of the disease seemed to vanish each time researchers thought they had found a lead. Some samples showed traces of viral RNA, but the sequences were fragmented like torn pages from a book. Others suggested bacterial involvement, but antibiotics proved useless. It was as though the illness was a ghost, killing without leaving behind the evidence needed to identify it. Years passed, and the outbreak eventually slowed, leaving behind a trail of graves and unanswered questions. Families built memorials, cities erected monuments, and officials promised investigations that never seemed to deliver. The survivors carried scars, both physical and emotional, that refused to fade. For them, closure meant knowing what had stolen their children, their parents, their partners. But the truth remained elusive, a wound left open by science's failure to provide answers. Then came the breakthrough. It did not arrive with fanfare or dramatic announcements, but in the quiet hum of a genetic sequencing laboratory. Advances in DNA technology had finally reached a point where degraded samples, once impossible to decode, could be reconstructed piece by piece. A small team of virologists decided to revisit the old samples, the very ones that had confounded the world years earlier. Using new high-resolution sequencing machines, they began to stitch together fragments of viral code from the remains of those who had perished. Slowly, like a puzzle forming from scattered pieces, a genome emerged. At first, the results shocked even the most seasoned researchers. The sequences revealed a virus unlike anything they had cataloged before. A hybrid carrying traits of multiple viral families, as though nature itself had stitched it together from different sources. It was no ordinary influenza, no simple fever. It was something new, something that had likely lingered unnoticed in an animal reservoir for decades before jumping to humans. The discovery was chilling. The ghost had a name, and it had finally been dragged into the light by science. The revelation was more than academic. With the genome mapped, researchers could now trace its origins, study its mutations, and, most importantly, begin to understand why it had killed so many so quickly. For the families who had waited so long, it meant the first glimpse of closure. For the world, it meant preparing for what could come next. The past had already claimed 100,000 lives. The future depended on ensuring it never happened again. When the genetic code of the mysterious virus finally appeared on computer screens, it was like staring at a fingerprint left behind at the scene of a mass crime. For years, the virus had mocked scientists, refusing to be identified. Now, with the genome assembled from broken fragments, investigators could at last begin tracing its story. The first question was obvious: where did it come from? To answer that, researchers turned to the growing databases of viral DNA and RNA sequences collected from animals, humans, and environmental samples across the world. Every virus carries markers of its ancestry, and these markers can reveal connections hidden in the tangled branches of evolution. The comparisons revealed a chilling truth. Parts of the new genome closely matched viruses found in bats, known carriers of countless pathogens. Other sections bore resemblance to strains of coronaviruses and paramyxoviruses, both families infamous for their ability to leap between species. This hybrid nature explained why the illness had been so difficult to identify in the early days. It was not simply one known virus acting in a new way, but rather a unique combination that had crossed into humans after decades of silent circulation in animals. The initial jump may have been rare, a single event in a market, a cave, or a remote village, but once inside the human body, it adapted with terrifying speed. As scientists examined its mutations, they found evidence that the virus had been evolving unnoticed long before the outbreak began. Archived samples taken years earlier from patients with mystery fevers were reanalyzed. To their astonishment, faint traces of the same genome appeared in blood from small outbreaks that had once been dismissed as isolated or unconnected. In hindsight, these were warning shots, early sparks that flared and died before exploding into the full inferno that claimed over 100,000 lives. The world had been staring at shadows of this virus for years, but had failed to recognize its potential danger. The story grew darker as researchers traced its path into humans. Environmental DNA collected from animal habitats showed strong evidence of the virus in bat populations. More disturbingly, fragments also appeared in samples from livestock pens near rural communities. It suggested that the jump into humans had likely been through an intermediate host, an animal caught between the wild and the domestic world. People living near these animals, handling them, eating their meat, or trading them in crowded markets, became the bridge. The virus had not appeared suddenly. It had been quietly building, preparing, waiting for the right moment to erupt. The investigation also revealed why the disease was so deadly. Unlike many viruses that settle into a balance with their hosts, this one was ruthless. Its hybrid genetic structure gave it multiple tools for survival. It could slip past the immune system, triggering delayed reactions that left the body unprepared. It attacked the respiratory system with the aggression of influenza, but also invaded blood vessels and organs, leading to multi-system failure. Some patients suffered strokes and heart attacks, while others experienced neurological collapse. The sheer unpredictability of its attacks made treatment nearly impossible in the early days. Doctors were fighting an enemy they could not see clearly, let alone understand. The DNA revelations painted a grim picture, but they also offered clarity. By sequencing the genome, scientists could now identify the exact markers of infection. Diagnostic tests that once produced inconsistent results could now be recalibrated for precision. More importantly, the genetic map allowed vaccine researchers to target the virus's weak points, the parts of its code that could not easily mutate without losing function. It was a scientific weapon, one forged too late for the victims already lost, but vital for the battles ahead. For families who had buried their loved ones without answers, the findings carried emotional weight. They learned that it had not been negligence, conspiracy, or divine punishment that stole their mothers, fathers, children, and friends. It had been a hidden predator, a virus lurking in nature, waiting for the chance to strike. The grief remained, but knowing the truth offered a measure of peace. Governments issued formal acknowledgments, admitting that their systems had been unprepared, that warning signs had been ignored. Memorials were held again, this time with names spoken alongside the virus that had taken them. The nameless killer had been unmasked, but the discovery also raised unsettling questions. If this virus could remain hidden for so long, how many others were waiting in the shadows? How many more creatures in forests, caves, and marketplaces carried seeds of the next catastrophe? The sequencing breakthrough was a victory, but it was also a warning. Humanity's relationship with the natural world had created a fragile boundary, one that could be crossed at any time. As scientists continued to work, one truth became undeniable: the past was no longer a mystery, but the future carried the same danger. The virus had revealed itself through DNA, but it had also revealed humanity's vulnerability. The challenge ahead was not just to understand what had happened, but to prepare for what might come again. The revelation hit the world like a thunderclap. For years, the mysterious outbreak had been a wound left open, a scar across nations that had buried over 100,000 lives without knowing what force had struck them down. When the DNA sequencing confirmed the virus's identity, governments could no longer hide behind uncertainty. The truth was undeniable. Humanity had been blindsided by a hybrid virus that had slipped silently from animals into people, spreading destruction before anyone even recognized its existence. The announcement was broadcast across television screens, news apps, and social media platforms. Headlines screamed of betrayal by nature, of scientific failure, of political negligence. For ordinary people, the news brought mixed emotions. On one hand, there was relief in finally having a name for the killer, finally knowing what had destroyed their families and communities. But that relief was braided tightly with anger. Why had it taken so long? Why had scientists ignored smaller outbreaks years before, outbreaks that in hindsight had been clear warnings? Families of victims staged demonstrations, demanding accountability from health authorities, accusing governments of failing in their duty to protect citizens. Protesters held pictures of lost loved ones, chanting that their deaths had not been inevitable but preventable. The virus was a natural enemy, but human negligence had made its rampage worse. Politicians scrambled to manage the fallout. In parliamentary halls, congressional hearings, and presidential offices, leaders were grilled about the early response. Documents were unearthed showing that reports of unusual fevers in rural regions had been filed and forgotten, brushed aside as irrelevant. Funds for research on emerging diseases had been cut in some nations, leaving laboratories understaffed and ill-equipped to study the mystery in time. International agencies were caught in the storm as well, criticized for being too slow, too bureaucratic, too cautious when action was needed most. The virus had not just killed, it had exposed cracks in the global system. The anger soon turned into a political consequence. Governments that had downplayed the outbreak lost credibility. Elections in several countries shifted dramatically, with health policy becoming the central battlefield. Candidates promised sweeping reforms, pledging that never again would a virus go undetected. Billions were suddenly funneled into public health systems, into genomic sequencing programs, and into laboratories tasked with monitoring every whisper of a new outbreak. The tragedy of the past had forced a revolution in the present. But it was not just politics that shook under the revelation. It was international trust. Some countries accused others of hiding early cases, of failing to report suspicious clusters, and of allowing the virus to spread unchecked until it crossed borders. Diplomatic meetings grew tense, with accusations traded like weapons. The World Health Organization was pressured to expand its authority, to send inspectors into nations whether they wanted them or not. Sovereignty clashed with survival, and the arguments revealed how fragile global cooperation truly was when confronted by invisible enemies. In the scientific world, however, the reaction was different. Laboratories celebrated the sequencing breakthrough as a triumph of perseverance and technology. Teams that had struggled for years against fragmented data now had a complete genome to study. Journals filled with papers analyzing every corner of the virus, mapping its proteins, its vulnerabilities, and its terrifying efficiency. For researchers, the revelation was not an ending but a beginning, a chance to finally fight back with knowledge instead of guesswork. Clinical trials for targeted vaccines were launched with urgency. Antiviral treatments were designed to exploit the virus's genetic weak spots. What had once been a ghost was now a blueprint for war. For survivors, the moment was deeply personal. Many had lived through the outbreak, but carried lasting damage, scars on their lungs, neurological problems, weakened immune systems. For them, the announcement was bittersweet. They could finally point to the culprit, tell their children the exact name of what had almost killed them. Yet, knowing did not erase the pain of living with what it left behind. Support groups swelled, connecting people across countries, united by loss and resilience. They shared stories, honored the dead, and pushed governments to remember that the tragedy was not just numbers but faces, families, and futures cut short. The media fed on every angle. Documentaries were filmed, journalists dug through archives, and whistleblowers came forward with stories of warnings ignored. The narrative of the nameless killer transformed into the virus we could have stopped sooner. Talk shows debated whether humanity's constant expansion into wild habitats had triggered the spillover. Environmentalists argued that deforestation, mining, and global trade were all complicit in the tragedy. The virus, they said, was not a freak event but the natural consequence of human greed pressing too far into nature's domain. Despite the outrage, one truth became clear. The DNA revelation had given humanity a weapon it never had before. The virus was no longer a phantom hiding in shadows. It had a genome, a history, and an identity. And with identity came power, the power to predict, to prepare, and, perhaps, to prevent the next catastrophe. But as the world celebrated this scientific milestone, a darker whisper lingered in the background. If one virus could kill 100,000 before being identified, how many more were waiting? The revelation of the virus's DNA sequence set off a global race unlike any in recent history. Overnight, the world's laboratories, pharmaceutical giants, and government health agencies shifted into high gear, determined to prevent history from repeating itself. The death toll of over 100,000 had left scars too deep to ignore, and now that the killer had a face, genetic, molecular, undeniable, the fight could begin in earnest. But the challenge ahead was monumental. This was not an ordinary virus, and fighting it required new thinking, new tools, and an unprecedented level of cooperation. The first priority was vaccine development. With the genetic blueprint finally in hand, scientists began analyzing the virus's spike proteins, the outer structures it used to infiltrate human cells. These were the keys that unlocked the body, and vaccines would need to block them. The hybrid nature of the virus made the task complicated. Its proteins carried features from multiple viral families, making it more adaptable than most traditional vaccine methods, using weakened or inactivated versions of the virus, were too risky and too slow. Instead, researchers turned to cutting-edge platforms, mRNA vaccines that could be designed rapidly based on the DNA code itself, and viral vector vaccines that carried fragments of the genome into cells to train the immune system. While vaccines were in development, pharmaceutical companies focused on antivirals. Genetic analysis had revealed specific enzymes the virus relied on to replicate. Targeting these enzymes became the strategy. If scientists could block the virus's ability to copy itself, they could stop it from spreading inside the body. Laboratories screened thousands of compounds, from brand new drugs to repurposed medications once used for other diseases. Some showed promise, binding tightly to the virus's weak points, slowing its devastating advance. For the first time, doctors had hoped that the next wave of infections, should it come, might not be unstoppable. But science was not the only front. Governments recognized that this was no longer a matter of health alone. It was a matter of national security. The virus had exposed just how fragile global defenses truly were. Leaders called emergency summits, pledging to create rapid response teams equipped with sequencing machines, mobile labs, and stockpiles of medical supplies. International databases were expanded to share genetic information instantly, ensuring that no unusual outbreak anywhere in the world could be ignored again. Some nations even established virus watchtowers, permanent surveillance stations in forests, caves, and animal markets, collecting samples to hunt for hidden pathogens before they spilled into human populations. Still, there was an unease hanging over the effort. The very nature of the virus, its hybrid genetic makeup, raised unsettling questions. Had it formed naturally, through years of cross-species mixing in the wild? Or had human activity accelerated its evolution by forcing animals into contact they would never have had before? A more disturbing theory circulated quietly among scientists and politicians alike, that the virus's strange genetic structure looked almost too deliberate, as though it had been shaped in a laboratory. Most dismissed this as conspiracy, but the whispers never fully disappeared, feeding public suspicion and distrust. For survivors and families of victims, the progress offered bittersweet comfort. Support groups watched the announcements with cautious optimism. They had waited years for answers, years for closure, and now they were being told that vaccines and treatments might be ready within months. Yet many asked the same haunting question: why hadn't this urgency existed when their loved ones were still alive? Why had it taken death on such a scale to push the world into action? Scientists acknowledged the truth. The tools now available had not existed a decade earlier. DNA sequencing technology had matured too late, but to grieving families, that was little consolation. In the background, the virus's discovery fueled a cultural shift. Documentaries flooded screens, telling the story of the nameless killer finally unmasked. Schools updated textbooks to include the tragedy, ensuring that future generations would not forget. Environmental activists used the crisis as proof of humanity's dangerous relationship with nature, demanding limits on deforestation, stricter wildlife trade laws, and investments in ecological balance. Economists, meanwhile, calculated the staggering financial cost of the outbreak. Billions lost in trade, productivity, and healthcare. The conclusion was clear: ignoring pathogens until they erupted was far more expensive than investing in prevention. As the months passed, the first human trials of vaccines began. Volunteers stepped forward, some motivated by hope, others by grief, determined that no one else should endure what their families had. Results trickled in. The immune systems responded, antibodies formed, defenses strengthened. The world held its breath as the data grew. Finally, it seemed, humanity was one step ahead of the ghost that had haunted it for years. And yet, even in victory, there was fear. The genome had revealed something scientists could not ignore: the virus was capable of mutating rapidly. While vaccines could protect against the current strain, there was no guarantee that future versions would not evade defenses. Nature was always moving, always adapting, always one mutation away from turning the tables. This realization created a grim resolve. Humanity could no longer think of outbreaks as isolated tragedies. They were inevitabilities, and the only question was whether the world would be ready the next time. The story of the virus, finally identified through DNA, became more than just a tale of loss. It was a warning written in blood and grief, urging humanity to remember that the line between safety and catastrophe is fragile. Science had finally caught up, but the war against what lurked in nature was far from over. Years after the virus was finally identified through DNA sequencing, its shadow still lingered across the world. More than 100,000 lives had been lost, and even though vaccines and antivirals were eventually developed, the scars of the outbreak ran deep, scars etched into families, communities, economies, and the very fabric of global trust. Science had answered the question of what, but humanity was left grappling with the far heavier question of why. The trials of pharmaceutical companies and health officials began almost immediately after the crisis stabilized. Families of the victims, organized into advocacy groups, demanded accountability. They argued that early warning systems had been ignored, that profit motives had delayed research, and that bureaucratic caution had cost lives. Class action lawsuits flooded the courts. Some corporations admitted no wrongdoing but quietly paid settlements to grieving families. Others stood firm, insisting they could not be blamed for a virus so unusual and so elusive. The truth lay somewhere in between. While no one had engineered the outbreak, the global health system had been unprepared, underfunded, and unwilling to invest in prevention until it was too late. The emotional toll was even harder to measure. Survivors continued to battle the virus's aftereffects: scarred lungs, neurological problems, weakened immune systems. They lived with daily reminders of what had nearly destroyed them, and many struggled with survivors' guilt, asking why they had been spared when so many others had not. Mental health crises surged in the years following the outbreak. Entire communities built memorials not just as places of mourning, but as warnings never again. In one city, a towering monument of steel and glass listed the names of the dead, lit at night so that the glow could be seen for miles. In another, a garden was planted where families gathered each year, both to grieve and to remind their governments of the price of negligence. Politically, the legacy of the virus reshaped entire nations. Health policy became central to elections, and leaders who had fumbled their responses were cast out of office. International alliances formed around the idea of pandemic defense, with countries pooling resources for surveillance systems, rapid response teams, and research hubs dedicated to emerging pathogens. Global trust, once fractured, began to heal under the realization that no country could stand alone in the face of invisible enemies. Yet suspicion still lingered. Nations accused of hiding early cases continued to face diplomatic isolation, their reputation stained by the belief that secrecy had worsened the tragedy. Culturally, the outbreak left behind an unease that never fully faded. Documentaries, films, and novels immortalized the events, turning the nameless killer into a grim chapter of modern history. Schools taught students about the virus as both a scientific case study and a moral lesson in accountability. Phrases like hidden hybrid,